Hello, everybody. Today we're going to look at MCAT biomolecules, and we're going to focus on water and fat-soluble vitamins. What a high-yield topic, vitamins. Everybody should understand vitamins, especially if you're going to be a doc. Today we're going to look at some of the biochemical mechanisms behind vitamins and show you that things like B12 and folic acid are interrelated. So we'll look at water-soluble. We'll look at B1, which is thiamine, B6, known as pyridoxal, B7, biotin, B12, also called cobalamin, vitamin C, of course, is ascorbic acid, and the fat-soluble vitamins are A, D, E, and K. So B12 is known as cobalamin. It functions in two biochemical reactions. One of them is methionine synthase. It's a cofactor in the synthesis of methionine from homocysteine. We're going to focus on that reaction because that's going to reveal the intimate relationship between B12 and folic acid. Reaction number two is methylmalonyl-CoA mutase. This is an enzyme that's involved in the actual degradation of odd-chain fatty acids as well as other amino acids. We're not going to elaborate on that very much. So over here is the structure of B12 determined by Nobel laureate Dorothy Hodgkin in the 1950s. What you see is the corin ring with 4 pyrrole groups. Notice pyrrole is a 5-carbon ring with it's a 5-membered ring with a nitrogen heterocycle. It's going to be linked to cobalt. Over here, we see cobalt has 6 ligands. We see above is X. X can be a methyl group. If it's the cofactor for methionine synthase, it can be a 5' deoxyadenosine if it's the cofactor in methylmalonyl-CoA mutase. But the actual cobalamin that you buy from the store is called cyanocobalamin, and the cyanide is actually a byproduct of the purification that is used to generate this. From B12 is only made in bacteria, so they grow up a lot of bacteria and they purify it on charcoal columns that actually have cyanide as a contaminant. Now, the cyanide you get from cyanocobalamin should not be considered a big health risk because you can't consume enough of it for the cyanide to be at any kind of level that would harm you. So B12 is intimately linked to the GI tract. So we consume B12 in food. We eat animal products. Now, I mentioned that animals don't synthesize B12. It's only synthesized by bacteria and some archaea. It's present in some plants because the microbes have an interaction with the plants, but the plants don't synthesize it de novo. So the B12 is stuck to protein. You actually chew that in your mouth, and then the salivary glands secrete haptocorin, or the R protein. The R travels to the stomach, and in the stomach, it's an acidified environment. And what happens is that the protein bound to B12 gets degraded, and so then the R protein binds to it in the stomach. But the stomach's parietal cells also synthesize intrinsic factor, or IF. And what happens is that as the chyme is passed to duodenum, the pH rises to a more alkaline level due to the pancreas's significant amount of synthesis and release of bicarbonate. And then at that alkaline pH, B12 binds intrinsic factor, where it shuttles it off to the ileum, and then the ileum has receptors that take it in by receptor-mediated endocytosis, where it's delivered to the actual liver itself. It's important to note that B12 is actually unstable in acid, and so it's always coupled to something that's chaperoning it around. So B12 is linked to tetrahydrofolate. I may say folate, but tetrahydrofolate is the active version of vitamin B12. Here's its structure right here. And so what happens is that 5-methyl tetrahydrofolate donates a methyl group, 1 carbon transfer, over to B12 to make methylated B12. And then the methylated B12 passes that methyl group onto homocysteine to make methionine, because methylated B12 is a cofactor for methionine synthase. So methionine is not an essential amino acid because we synthesize it, but we can find out that in B12 deficiency, it becomes essential. So tetrahydrofolate exists in multiple forms. I mentioned 5-methyl tetrahydrofolate, but we have other forms too. Here's the formal form. So formals, one carbon there. There's methylenol, the double bond. There's methylene, the CH2, and the 5-methyl. All these steps are interconvertible with the exception of the conversion of methylene to 5-methyl tetrahydrofolate. This irreversible step gives rise to something known as a folate trap, which I will elaborate on in a moment. But each one of these forms of tetrahydrofolate can be a cofactor for a different enzyme. For example, formal tetrahydrofolate is used to actually transfer formal groups to transfer RNAs. Then methylene tetrahydrofolate is a cofactor for thymidylate synthase, which converts uracil into thymine, and then 5-methyl THF, as I mentioned, is involved in the synthesis of methionine. So what about B12 deficiency? Normally our livers can store vitamin B12 for a few years, but there are some ways you can deplete it. One way that's common in older people is they have a condition known as pernicious anemia. This is an autoimmune disease where you attack the parietal cells in the stomach and they can no longer synthesize intrinsic factor to bind B12. As a consequence, you become B12 deficient. So you can be B12 deficient despite the fact you take in the vitamin as a supplement every single day. So what happens when you're B12 deficient? So you can probably predict a few things are going to happen. Well, one of them is going to be that you can no longer transfer the 5-methyl THF to B12. And so this THF is no longer liberated. And so many of these forms of folic acid could no longer be generated. So B12 deficiency equals folic acid deficiency. And so you can also predict that what's going to happen is that methionine synthase won't be in business anymore. So you elevate the methyl THF, but you also elevate homocysteine levels here. So you can actually further elevate homocysteine because you can consume methionine and protein in your diet. And you can have S-adenosyl methionine and S-adenosyl homoserine actually make this over here. So you can elevate homocysteine levels even further despite the fact that methionine synthase is deficient. And this has caused a number of problems that are cardiovascular and neurological in nature. And so homocysteine, when it's elevated, is pretty bad news and needs to be clinically treated. There's also a disorder known as methylene tetrahydrofolate reductase, or MTHFR deficiency. As you can see over here, if this enzyme is deficient, you'll no longer synthesize 5-methyl THF. And so what does that do? It disallows for vitamin B12 to be methylated. As a consequence, what happens is that you're going to have a difficult time making the other folic acid vitamers. And so as a consequence, you also elevate homocysteine as well. So the trick to solving this problem is to actually supplement your diet with 5-methyl THF. What you can see here is if you took formal THF as a supplement for this deficiency, you would never be able to generate it because the enzyme is missing. So what we see then is that folic acid or tetrahydrofolate is intimately linked to the chemistry of vitamin B12. Folic acid is also involved in DNA synthesis. And so what we see here is the conversion of uracil into thymine by dihydrofolate reductase, which uses TH4 as a mechanism. And as a consequence of adding the methyl group, what you do is you reduce TH4 to TH2. In order to get it back into business as TH4, you have to actually use dihydrofolate reductase. So it turns out, though, that methotrexate, a well-known, well-established cancer drug, it actually inhibits dihydrofolate reductase. So methotrexate is called a folate antagonist because it looks like a substrate. Now, what happens in this case is that these cells, since they don't have the capacity to synthesize DNA, what they do is they grow really big in G1, and they're called megaloblasts. You actually get megaloblastic anemia as a consequence of the inability to synthesize DNA, which is derived from either folic acid or B12 deficiency. We switch over to pyridoxal phosphate. The pyridoxal, the A-al means aldehyde. Here you see the aldehyde group here. This is an amazing cofactor, or vitamin B6, it catalyzes hundreds of reactions. And what you see on the right panel up here is that this aldehyde links to an enzyme's lysine residue to generate a Schiff base, also known as an imine. It does a number of things to amino acid substrates, including decarboxylation, transamination, racemization, as well as deamination. I'll show you a really important one known as transamination, as you see here. And what does transamination do? So when you want to metabolize or catabolize amino acids, what you do is you got to get rid of the nitrogen ammonia group. Now, the way you do this is you transaminate. So the way you get rid of it is you put it on to a substrate like alpha-ketoglutarate to generate glutamate, which means that glutamate is a carrier of amino acids. And we discussed this when we were talking earlier on about amino acid chemistry. Now, the phenylalanine then is converted into its cognate alpha keto acid, which is phenylpyruvate, and this is coordinated by the action of transaminases. These are clinically important because alanine transaminase, or ALT, is one of the liver enzymes that docs take a look at when they're concerned their patients have problems with their liver, like cirrhosis or cancer or something else. So ALT is something you would order as a clinical lab. We move over to biotin. Biotin is vitamin B7, and this is a really interesting one because it's involved in carboxylation, so it adds CO2 groups. So it is adding carbons to substrates. Here you see the acetyl-CoA carboxylase enzyme uses biotin as a cofactor to add a CO2 to acetyl-CoA to make malonyl-CoA. As this is the rate-limiting step in fatty acid synthesis, it's promoted by insulin, but inhibited in the liver by glucagon. Notice that the CO2 is derived from bicarbonate and it uses ATP. So here's the Biotin cycle. So this is pretty interesting. So here's the free biotin pool. The biotin has not been activated. There's no carboxyl group to donate anything to anybody. The free biotin pool, here's biotin existing with this long arm with a carboxyl group. And apo enzymes are converted into holo enzymes by covalently linking this carboxyl side chain onto a lysine group from the enzyme. And that generates an amide linkage because you know a primary amine, i.e. lysine, plus a carboxylic acid generates an amide. And so these holo enzymes then can actually use ATP and add on the carboxyl to load up the biotin, and that activated carboxyl is going to be used to donate as a substrate to fatty acid synthesis enzymes like acetyl-CoA carboxylase, to gluconeogenic enzymes like pyruvate carboxylase, which makes oxaloacetate, as well as the catabolism of some amino acids. And so once the biotin has been activated and donated its carboxyl group, then what happens, it gets reconverted into the free biotin pool. And at this point, dietary biotin that you may have consumed is also integrated into this step here. And this generates biocytin, and biocytin is hydrolyzed back into the free biotin pool by biotinidase. As you can imagine, it hydrolyzes this peptide bond there. So we go over to thiamine, or vitamin B1. So we'll see a cascade of reactions that's best illustrated by the action of the pyruvate dehydrogenase complex. This mitochondrial complex converts pyruvate, a 3-carbon entity, into the 2-carbon acetyl-CoA and liberates CO2. And during that oxidation event, we not only create NADH, but we also create acetyl-CoA, which I'll show you is an activated 2-carbon thioester. An analogous reaction occurs in the Krebs cycle, where you convert isocitrate, a 6-carbon entity, into the 5-carbon intermediate alpha-ketoglutarate. You kick off a CO2. Whenever you make CO2, the highest oxidized form of carbon, you also make reducing power, as you see there. So what does thiamine do? It's involved in breaking carbon-carbon bonds, and that's what we have to do to make CO2. So thiamine is the cofactor. It's right here. what you see is a nitrogen that's positive next to a carbon that's negative. Whenever you have a negative atom next to a positive atom, it's called an iliad, Y-L-I-A-D. This is a plus next to a minus. It's not a book written by Homer about Odysseus. Rather, what we see over here is pyruvate. I've labeled the actual carbons so we can follow them through the cycle here. So what happens is the carbanion attacks an electrophilic carbonyl, generates a tetrahedral intermediate that when it collapses, these electrons are pulled towards this nitrogen. Positively charged nitrogens are often thought of as electron sinks. So as it pulls those electrons towards itself, it liberates CO2. And so one of the carbons is going to be liberated as CO2. And now what we have are the activated 2 carbons from that event here that are stuck to the thymine. And we're going to go through a long road to grandma's house to actually regenerate this thymine here. And what we do is we use lipoic acid. Here's lipoic acid right here. This negative carbanion attacks one of the sulfur groups, lipoic acid. And if you think about lipoic acid a little further, what you recognize is that it has 8 carbons. It's actually derived from the breakdown of fatty acids. This is octanoic acid derivative right here. And so lipoic acid has a really long, here's this carboxyl group here. It can link up to lysines through amide linkages as well to make lipoyl lysine. It's reminiscent of what we saw with biotin. And so enzymes that use lipoic acid are actually covalently linked through their lysine residues as well. And so what happens then is that this carbanion attacks the enzyme linked lipoic acid. You make your tetrahedral intermediate. You pull off a proton. You generate the oxygen anion right here. And what you do is you liberate the original thiamine. Okay, here's the iliad again. And so, but you're stuck with the 2 carbons that were initially in pyruvate. Now there's 2 carbons and it's linked to lipoic acid. And this linkage is a thioester. So what you should recognize is that thioesters are high energy bonds that have the same amount of energy released upon hydrolysis that phosphoanhydride bonds have. So what we see is that the 3 carbons that were initially in pyruvic acid, one of them is now CO2, and that oxidation event is going to liberate a lot of energy, but we've captured some of it in the form of the thioester bond, because remember, energy can never be created, or it can either be created or destroyed, but it can be converted from one form to another. And now we see it's in the activated form of the thioester group. And what happens then is coenzyme A, which has a sulfur group itself, actually attacks this electrophilic carbon here. You kick out this lipoic acid. Now it's reduced, and you make acetyl-CoA. This is a trans-thioesterification reaction because we start out with a thioester here, and we end up with a new thioester. And so let's take a look at pantothenic acid real quick. This is vitamin B5. You can find it on the box of your cereal acid. It's basically the precursor to coenzyme A, which is this complicated structure over here. But the take-home message is that the key active component is the actual sulfhydryl group over here. And what we do then is we actually have, here you can see, basically it looks like acetyl-CoA. So then what we've done is we've made acetyl-CoA, but we have reduced formal lipoic acid, and we use NAD to generate it back in to the oxidized form. So future rounds of activated thymine can be used in the pyruvate dehydrogenase complex, which is shown here in all of its complex steps. There's a lot of chemistry going on here. We see decarboxylation by thymine, we see lipoic acid, we see coenzyme A, as well as NAD. And so we switch over to vitamin C, and vitamin C, as we all know, is ascorbic acid here, and ascorbic acid can be converted into dehydroascorbic acid by a redox reaction. Here, these electrons are released, and as a consequence, what you can do is couple it to the conversion of oxidized glutathione into reduced glutathione. Here you see glutathione is a tripeptide, and what it has is actually a sulfhydryl cysteine residue here, that could be converted from the oxidized and the reduced form. Here you see the cysteine linkage here. Vitamin C is actually a dicarboxylic acid. And if you think about vitamin C closer, it's actually derived from hexoses. And in fact, vitamin C gets into cells through glucose transporters, which is pretty interesting. So it's a dicarboxylic acid. The first pKa is 4.1. What does that mean? It means that at physiological pH of 7.4, that's going to be virtually 100% ionized into the ascorbate form. But the second pKa is 12. And so that's probably not going to exist at any significant amount at physiological pH. But what ascorbate can do is it can actually be involved in antioxidant reactions, because what you see here is that ascorbate can actually donate an electron to a free radical to neutralize it, which is a good idea. And the ascorbate radical that's generated here, as you might guess, is fairly well stabilized by the electron density in that ring itself. So ascorbate donates electrons to free radicals to neutralize them. And here you see a couple of reactions that vitamin C performs. Dopamine to norepinephrine. So what you can do is add a hydroxyl to this catecholamine here to generate norepinephrine. What's a catecholamine? Well, a catecholamine has an amine function, a benzene ring, and two hydroxyls that are in the adjacent ortho positions to each other. So this is conducted by a monooxygenase. We also see that the conversion of proline, the hydroxyproline, and lysine to hydroxylysine, this redox reaction that modifies collagen, is also facilitated by vitamin C itself. And in this case, it's involved in converting the ferric form of iron to the ferrous form of iron. So now we'll switch over to the fat-soluble vitamins, A, K, E, and D. And our first one up is going to be vitamin A. And so what you see over here, vitamin A looks like a pretty hydrophobic compound. It's going to be fat-soluble. And so all these hydrophobic fat-soluble vitamins, they need binding proteins. They need somebody to carry them around because they'd precipitate like a rock in the serum. You should also recognize that vitamin A is composed of these 5 carbon isoprenoid units. These are things that are synthesized in the methylonic acid pathway, which is really the pathway that cholesterol uses. So we see the ester form, we see the retinol, which is vitamin A1, the alcohol form. You can oxidize this alcohol to retinal, to the aldehyde, which can be further oxidized into retinoic acid. These are all vitamins of vitamin A, but we consume beta carotene, which is a provitamin, is converted in the intestine to these forms, and it's the retinol esters that are actually incorporated into the chylomicrons. We saw the chylomicrons earlier when we discussed the actual fed state. And so chylomicrons are synthesized in the intestine, they're transported to the lymph and blood, and ultimately the destination for the retinol ester is going to be in the liver. But when the liver wants to release it, it will release it as retinol, and then the retinol binding protein comes in and shuttles it off to target tissues. And one of the major function of vitamin A is actually in gene expression. Although, yes, it does have a role in vision. That's its most notorious form. It's not its major form, though. In the visual cycle, 11-cis retinol is converted to transform by light. And what you can see here is that the transretinal, the aldehyde, is actually covalently linked to a protein called opsin. So we have an aldehyde plus lysine giving us an amine. We see lysine involved again to generate a protein known as rhodopsin, which is involved in the visual cycle. Vitamin K. Vitamin K has a number of vitamins. There's K1, K2, and K3. It's incredibly hydrophobic. As you can see, look at these long isoprenoid arms hanging off of it as well. Now, one of the key structural features is this quinone. Quinones basically are ring structures that have a carbonyl at the top and a carbonyl at the bottom. Those can be oxidized into alcohols and, reduced into alcohols. as you can see here, this alcoholic form of the quinone or vitamin K is involved in converting a precursor blood clotting factor glutamate residue. It carboxylates to an active clotting factor. And in this conversion, what you do is you generate this epoxide form of vitamin A. And in order to regenerate it, you go through this pathway diagram at the bottom here that involves some redox chemistry. And as a consequence, what you can do is inhibit this with drugs like warfarin. And what warfarin will do is prevent the actual clotting cascade from occurring. So warfarin's a blood thinner. So vitamin K is for vitamin clot. Now, vitamin E or tocopherols are pretty interesting. They're involved in free radical chemistry themselves. And you may have seen this as an ingredient in a number of foods. So when you go to the supermarket, look at the side of cereal boxes and other foods. And what it does is it's a preservative for a longer shelf life. So here's what it looks like. It's pretty hydrophobic as well. It's all a bunch of saturated carbon groups over here. Now, what you can see is that this LOO radical is going to be turned into a neutral radical by the action of tocopherol. So what's LOO radical? Well, it's actually lipid peroxide. So here's an unsaturated lipid in your membrane. It can be converted into a radical here. And then oxygen can generate a peroxy radical. Those are dangerous because what they can do is basically disrupt the structure of the membrane. So vitamin E is in the membrane, and what it does is it actually takes this free radical here, okay, and generates the neutral form of it, which is a good idea. So then this alpha tocopherol that has a radical, how is it neutralized? Well, one way is through vitamin C. So lastly, we're going to look at vitamin D. Vitamin D is incredibly interesting. Now, vitamin D isn't really a vitamin because A, we make it. It's really a corticosteroid. Why is it a steroid? It's derived from cholesterol. It's actually precursor 7-dehydrocholesterol. This is in your skin, okay? And when UV light hits your skin, there's a non-enzymatic reaction that rearranges and breaks some bonds, and you make cholecalciferol, which is going to find its way into the liver. And what the liver will do is it'll hydroxylate it, okay, to make 25-hydroxy cholecalciferol. That's called calcidiol because it has two alcohols. But in the kidney, what happens is that the parathyroid hormone will stimulate the synthesis expression of the alpha-125 hydroxylase to make the active form of vitamin D. This is vitamin D3 here. The active form of it is the 125-OH form. When you take vitamin D supplements, they're not in the active form. They actually have to be activated in the liver as well as in the kidney. And so just like vitamin A needs a binding protein because it's so insoluble, so does vitamin D. So our passage today will look at vitamin D binding protein, the knockout of it, and how it affects the actual vitamin D reactions. And what does vitamin D do? Of course, it stimulates the uptake of calcium and phosphate in the intestines is its major function. And so how does it do that? Well, it's actually involved in transcription. So it's really a hormone. So what happens is that vitamin D circulates in the serum. It's bound to its binding protein. But when it reaches its target cells, the binding protein disappears. And this steroid can diffuse through the membrane, interact with the vitamin D receptor, so it's like a steroid nuclear hormone, arrive in the nucleus, and it partners with the RXR transcription factor, where it conducts transcription programs that are involved in differentiation, proliferation, apoptosis, as well as the calcium metabolism, of course. So vitamin D is intimately linked to the parathyroid hormone. We have 4 parathyroids that are embedded in our thyroid. In response to low calcium, parathyroid hormone is released. It has multiple functions. Down here, we see its function on the kidney is to increase the expression of the hydroxylase that makes the 125 triol is what it's called, calcitriol. The names get confusing. You got to memorize these things and learn them. So what does this do? Well, in the kidneys, it actually preserves calcium, okay, by keeping it in the serum, and then it also kicks out in the urine phosphate. But over here, when it travels, it can affect the bone and the intestines by causing the intestines to increase more calcium absorption, but also parathyroid hormone works on the osteoclasts as well as the osteoblasts. So the clasts are involved in degrading bone. The blasts are involved in synthesizing and depositing the mineral that makes bone. And so if you're low in serum calcium, you can go to the bone bank, which is going to have the source of calcium. The osteoclasts will help release it from the actual bone itself to raise serum calcium levels. So what we understand then is that vitamin D, calcium, parathyroid hormone, are all intimately linked to bone structure. And so you have to think about bone structure when you think about vitamin D. Here's a sample of the bone inside the spongy form of it. Outside is the hard cortical structure, which is composed of the osteon. Here's an osteon here. What you see is the Herversian canal in the center, which has the blood, the lymph, and the nerves. But over here in the lacunae, which means pit, you have the osteoblasts, which also interact with the osteoclasts, which you don't see here. And it's the interaction between the osteoblasts and the osteoclasts that determines whether you're going to synthesize bone or whether you're going to degrade it with the osteoclasts. So with that in mind, let's do some cool workshop passages. They're going to challenge the boundaries of our knowledge, and hopefully we'll be able to apply our knowledge, our foundational content review knowledge, into experimental situations.